Welcome back to our flatworm series. Hi, I'm Christopher. This is the final cestode video in this short review series. And today we are cleaning up the organisms that did not fit neatly into the earlier videos. That does not make them random, it means the sorting clues change. Start with the host or clinical context, then ask what intermediate host or exposure closes the loop. Site, life stage, and diagnostic clues to finish the identification. By the end, every leftover gets a drawer, and the cabinet should make more sense than the alphabet soup. Here's the retrieval sequence for this final group. Start with the host or clinical context: horse, ruminant, bird, rodent, person, aquatic chain, or unsolved mystery drawer. Next, ask what intermediate host or exposure closes the loop: batted mite, arthropod, copepod, fish, a second vertebrate host, or a root that is still incompletely resolved. Once the host and exposure have narrowed the choices, use the parasite's location and life stage as a final check. The cabinet is simply a way to organize the biology. Get to the right group first, then use the details to identify the parasite. Here's a fast way to sort these out. An ore-batted mite is a tiny, free-living soil mite common in pasture. It becomes the intermediate host when it ingests cestode eggs. If the definitive host is a horse, think Anoplocephala perfeolata, especially around the ileocecal region. If the definitive host is a ruminant, think Menesia in the small intestine. Same pasture mite, different definitive host. The mite is moonlighting, and your job is to check which host signed its paycheck. For the bird drawer, think Raeliatina. Adults live in the small intestine, and the intermediate host is commonly an ant or beetle, depending on the species of tapeworm. The eggs are not operculated. They are packaged in egg capsules and contain hexacanth embryos with 6 hooks on the oncosphere. Proglottids may also be seen. The name Raeliatina honors Alcide Rayet, a French veterinarian and parasitologist. For the rodent-human drawer, think Hymenaleptis. H. diminuta and H. microstoma use arthropod intermediate hosts. Hymenaleptis nana is the special case because it can complete a direct life cycle and auto-infect. Hymenaleptis eggs are also non-operculated. H. nana has polar filaments, H. diminuta does not. Now we change the rules. Spirometra and the broadfish tapeworms follow Diphyllobothridian logic, traditionally taught under the older Pseudophilidian label. Think water, operculated eggs, a copepod first, and attachment grooves called bothria. A second vertebrate host usually follows, and the larval names suddenly matter. These are tapeworms with a suspicious amount of fluke energy. Spirometra adults live in the small intestine of dogs, cats, and other carnivores. Operculated eggs pass in the feces and must reach water. A coricidium hatches and is eaten by a copepod where a plerocercoid develops. But one intermediate host is not enough. Spirometra requires a second vertebrate host. When a non-fish vertebrate eats the infected copepod, the plerocercoid develops into a plerocercoid, commonly called a sparganum. That second host can come from almost anywhere in the vertebrate world, except fishes. It may be amphibians, reptiles, birds, or mammals. And now the life cycle gets interesting. Once a sparganum has formed, it can be passed through successive paratenic hosts. One vertebrate eats another, and the sparganum transfers to the new host, and no additional larval development is required. It can continue through the food web until a suitable definitive host, such as a dog or cat, eats the infected vertebrate. Then the sparganum finally develops into the adult tapeworm. People are accidental hosts. Drinking water containing infected copepods can introduce the earlier larval stage, while eating raw or undercooked infected tissues can introduce spargana. Migrating spargana in human tissues causes sparganosis. And remember, the adult tapeworm in the dog or cat is not a zoonotic threat. The aquatic life cycle still has to happen. The broadfish tapeworm uses a parallel aquatic chain. Operculated eggs reach the water, a copepod becomes the first intermediate host, and a fish becomes the second. A larger predatory fish may act as a paratenic host. Humans and other fish-eating mammals acquire the adult by eating infected fish that have not been adequately cooked or frozen. The naming of this parasite has changed. Many medically important species formerly placed in Diphyllobothrium are now classified in Dibothriocephalus, including D. latus, D. nihoncaense, and D. dendriticus. D. latus classically points towards freshwater fish. D. nihoncaense is associated with anadromous Pacific salmon, so raw salmon can be a transmission route. The risk depends on the parasite, not the menu font. Proper commercial freezing lowers parasitic risk, and cooking remains safest. Mesocestoides brings us back to cyclophilidian cestodes, but it refuses to follow a completely resolved map. Adults live in the small intestine of dogs, cats, and other carnivores. Egg-laden proglottids are shed, while free eggs are uncommon. For intestinal infection, proglottid morphology and the periuterine organ are the most useful clues than waiting for the abundant free eggs on flotation. The larval stage is called the tetrathyridium. Tetrathyridia may occur in body cavities or organs, and can multiply asexually, helping explain severe peritoneal and occasionally pleural disease. Keep the morphology clean: 4 suckers, no rostellum, and no hooks. The complete life cycle remains incompletely resolved, and the first intermediate host is uncertain. Mesocestoides is the drawer that files a missing person report on its own first host. Now turn the drawers into clinical reasoning. First, ask what stage could realistically appear in the sample you can collect. Fecal flotation can reveal Hymenoplectus and Menesia eggs. Aquatic forms produce operculated eggs, so concentration or sedimentation may be useful. For Menesia, Rayletina, and Mesocestoides, gross examination of proglottids and a proglottid crush can be particularly helpful. A negative equine fecal examination does not reliably exclude Anoplocephala perfilata because corpologic sensitivity is limited. Larval disease changes the diagnostic window. Spargana, plerocercoids, and tetrathyridia may require tissue examination, imaging, effusion cytology, surgery, histopathology, or molecular confirmation. Then return to exposure, or batted mites for Anoplocephala or Menesia, bird plus arthropod for Rayletina, rodent-human context for Hymenoplectus, freshwater copepods and vertebrate prey for Spirometra, raw or undercooked fish for broadfish tapeworms, and an unresolved first host for Mesocestoides. Control follows the life cycle. Treat definitive hosts appropriately, prevent predation and scavenging, avoid raw vertebrate tissues, use safe water, and cook or properly freeze fish. Control poultry arthropods where practical. Pasture mites are ubiquitous, so routine mite suppression is generally impractical. Let's close the cabinet one drawer at a time. Horse plus ore-batted mite, Anoplocephala. Ruminant plus ore-batted mite, Menesia. Same mite, different tapeworm, different definitive host. Bird plus ant or beetle, Rayletina. Rodent or human context plus an arthropod, or the direct H. nana exception, Hymenalepis. Water plus copepod plus a second vertebrate host carrying a sparganum, Spirometra. Water plus copepod plus fish, Dibothriocephalus, and the remaining Diphyllobothrium context. Carnivore plus vertebrate prey, tetrathyridia, and an incompletely resolved first host, Mesocestoides. Then choose the diagnostic clue that matches the host and life stage: eggs, proglottids, or tissue. Finally, control the exposure route that actually keeps the cycle moving. Organize the biology first, so each species name has a host, a life cycle bridge, and a diagnostic story to attach to. Shelf beat soup, patterns beat panic. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.